Hello students, this is Rekha from Government Science College. So today we shall discuss the factors affecting the rate of transpiration, antitranspirants and guttation. So the factors affecting the rate of transpiration can be categorized under two headings. First one is external or environmental factors and second one is internal or structural factors. The external factors include humidity of the air, temperature, wind velocity, light, water supply, atmospheric pressure, sprays and dusts, and vital activities. The internal or structural factors includes the stomatal aperture and its frequency, the water content of the mesophyll cells, and structural peculiarities of the leaf. So first coming to the external or environmental factors. So all those environmental factors which affect the diffusion pressure deficiency gradient affects the rate of transpiration. Firstly, the humidity of the air. The relative humidity of the atmosphere affects the rate of transpiration to a great extent. The higher the relative humidity of the atmosphere, lower is the rate of transpiration. The rate of transpiration increases with the decrease in the humidity of the external atmosphere. Second one is temperature. The increase in temperature increases the rate of transpiration by increasing the rate of evaporation of water from the cell surface and decreases the humidity of the external atmosphere. For every 10 degree raise in the temperature, the rate of transpiration it gets doubled. Next, the third one is the light. Light has got a direct as well as an indirect effect on the rate of transpiration. So light affects directly by increasing the rate of transpiration and indirectly in two ways. Firstly, by controlling the stomatal opening and secondly by affecting the temperature. With the increase in the light intensity, the rate of transpiration increases because the stomata get opened and the temperature raises. The rate of transpiration increases markedly in the light and decreases in the dark. There is a close relationship between the opening of stomata and presence of light. Next, coming to the soil water. When water is less in the soil, plants begin to senesce, that is, premature aging takes place, which results in the fall of the leaf and hence the transpire and hence it transpires less amount of water. Next is the wind velocity. The increase in the wind increases the rate of transpiration by removing the water vapor of the atmosphere and lowering the relative humidity. The transpiration is faster in mild wind. If the wind is of high velocity, then it retards the rate of transpiration, probably because the guard cells become flaccid and the stomata are closed. So next is atmospheric pressure. The reduction of atmospheric pressure reduces the density of the external atmosphere, thus permitting more rapid diffusion of water. So thus the plants growing on hills show higher rates of transpiration because of low atmospheric pressure and they develop xerophytic characters. So next is sprays and dusts. So sprays and dusts affect the permeability of the cuticle and temperature of leaves. For example, the Bordeaux mixture, which is an antifungal agent, it lowers the leaf temperature and increases the permeability of the cuticle, thus increasing the cuticular transpiration, especially during night. So next one is the vital activities. Vital activities of the plants may also affect the rate of transpiration, such as the heat produced during respiration increases the rate of transpiration. So these are some of the external or the environmental factors. Coming to the internal or the structural factors, first one is the number of leaves present in a plant. More leaves means a bigger surface for transpiration, more stomata, and hence more water loss. And the number of stomata also plays an important role in transpiration. More stomata means more pores for transpiration. Thirdly, the size of the leaf. A leaf with a bigger surface will transpire faster than a leaf with a smaller surface area. Presence of the cuticle. A waxy cuticle which covers the epidermis, it reduces the transpiration. It reduces the cuticular transpiration. And some of the leaves, they get modified into prickles, spines, scales, phyllodes, cladodes, etc., which reduces the transpiration. The mesophyll cells, the compact mesophyll cells, reduces the transpiration, while the loose mesophyll cells increases the transpiration. Next one is stomatal frequency. Stomatal frequency means the number of stomata per unit area of the leaf surface. So this stomatal frequency, it varies from plant to plant and also depends on the effect of environment. Salisbury used the term stomatal index to represent as I is equal to S divided by E plus S into 100, where I stands for stomatal index, S for number of stomata per unit area, and E for the number of epidermal cells in the same unit area. So increase in stomatal frequency increases the rate of transpiration. So this is about the factors which affect the rate of transpiration, both the external and the internal factors. So the next topic is plant antitranspirant. So what are these antitranspirants? See, in transpiration, almost 98% of the water absorbed by the plants is lost and only an insignificant amount of water is utilized by the plants for its own purpose. So due to this, plants have to face several problems. So if this loss of water is reduced, it will be an asset to the nature. So as a result, scientists have been investigating on these lines and they have found out certain substances which reduces the transpiration rate without adversely affecting the exchange of gases during photosynthesis and respiration, and these are called as antitranspirants. The term antitransparent is used to designate any material applied to the plants for the purpose of retarding transpiration. They are applied to the leaves, cut flowers, newly transplanted shrubs to preserve and protect the plants from drying out too quickly. So how does this antitransparence reduces the rate of transpiration? See, firstly, it reduces the absorption of solar energy and thereby reducing the leaf temperature and transpiration rate. and secondly by forming a thin transparent film which hinders the escape of water vapors from the leaves. Thirdly, by promoting the closure of stomata, thus decreasing the loss of water vapors from the leaf. So, a few examples of antitransparents are colorless plastics, silicone oils, low viscosity waxes. So, these are sprayed onto the leaves to form a thin film which is permeable to carbon dioxide and oxygen but not to water. And secondly, the phenyl mercuric acetate, and these helps in the partial closure of stomatal pores for at least 2 weeks. The abscisic acid induces the stomatal closure, and the carbon dioxide is also one of the effective anti-transparent. Its higher concentration cannot be used because it results in complete closure of stomata, affecting adversely the photosynthesis and respiration. But however, this carbon dioxide usage is not economical and it is practically feasible only in experimental glass houses. So the third topic of discussion is guttation. So guttation is the exudation of water from the plants in the form of liquid. The term guttation was proposed by a scientist by name Burgerstein. So the amount of water lost by this process is negligible when compared to transpiration. Guttation is not universally observed in all plants. It is restricted to certain genera, especially the herbaceous plants. So this guttation takes place usually early in the morning when the rate of water absorption and the root pressure are higher and the transpiration is very low. The plants showing guttation shows the presence of specialized structures called hydathodes which are present on the tips and margins of the leaves. So you can see the gutted water at the tip and the margin of the leaves in the diagram. So what does this gutted water contain? It contains the enzymes, sugars, amino acids, organic acids, vitamins, the organic compounds and some of the mineral salts. So this is the vertical section of the leaf which passes through the hydathode. So the plants showing guttation shows the presence of the specialized structures called hydathodes on the tips and margins of the leaves. Each hydathode consists of a pore in epidermis followed by a large intercellular space, loosely arranged parenchyma and the xylem elements, especially the tracheates. The loosely arranged mass of parenchyma is known as epithem. So there are two types of hydathodes, namely the epithem hydathode and active hydathode. Epithem hydathode is the one where the water is forced out by the root pressure. Active hydathode is the one where the water is secreted by the force developed within the cells themselves. So what is the mechanism of guttation? See, when the soil is flooded with water and the humidity is high in the atmosphere, the plant's root system absorbs excess amount of water. As a result, a hydrostatic pressure develops in the roots which forces the water upwards. The water involving other soluble cell components is forced out of the xylem present in the epithem tissue. As a result, stomata which are filled with the water is forced out by the root pressure. The liquid is forced out of the pores where the stomata offer no resistance. So coming to the major differences between guttation and transpiration. Transpiration occurs during daytime whereas guttation occurs during night time. Water is given out in the form of vapor during transpiration and in the form of liquid in guttation. The transpired water is pure, whereas gutted water contains dissolved salts and sugar. Transpiration takes place through stomata, lenticel, or cuticle, whereas guttation it occurs through the hydathode found only on the leaf tips or margin. Transpiration is a controlled process which lowers down the temperature of the surface, whereas guttation is uncontrolled process which lacks such relationship. So next is the differences between the stomata and the hydathode. Stomata occurs in epidermis of leaves and young stem, whereas hydathode occurs at the tip or the margin of the leaves. Stomatal aperture is guarded by two kidney-shaped guard cells, whereas the hydathode is surrounded by a ring of cuticularized cells. Two guard cells are generally surrounded by subsidiary cell in stomata, whereas it is absent in hydathode. Opening and closing of stomatal aperture is regulated by guard cells, whereas in hydathode the pores remain always open. And these are involved in transpiration. Stomata are involved in transpiration and exchange of gases, whereas hydathode it is involved in guttation. To summarize the topic, transpiration and guttation are vital and partly a physical process controlled by both internal and external factors. Guttate fluid contains various micro and macromolecules which can be collected and can be used in the production of recombinant proteins. The knowledge of plant antitransparents may find its use in crop production. Thank you for listening this lecture.